Welcome back, guys. Today we are continuing those complex patterns of inheritance that don't fit Mendel's observations. And we left off at X-linked inheritance patterns. So today we're going to be starting with something called multiple alleles. All right. So we get everything kind of squared away here. So this would be number 4 in our list. And we are talking about multiple alleles. Multiple alleles. So multiple alleles, obviously, multiple means more. So more than the traditional 2 that we've seen so far. So more than 2 alleles at a gene. And typically, when we think about multiple alleles, the classic example of this is blood types, where we have 3 alleles at work here. We're going to have allele A, which is going to convey a blood type A phenotype. We're going to have allele B, which is going to convey a blood type B phenotype, and these are co-dominant. We've talked about that last time. You can have blood type AB if you have both allele sets. And then our third allele is going to be this mutant allele, where if you get this mutant allele, you don't produce any antigen, and this is going to be recessive to blood types A and B. So we have this interesting allele set that we have. So you want to think of it like this. Remember this here, right, is a red blood cell, a RBC. And somebody with blood type AB is going to have both the A antigens. So we're going to sketch and draw some of these A antigens here. And then we'll also have the B antigens. So I'm going to show these B antigens here as well. And this would be somebody with blood type AB. They've got allele A and allele B on the 2 chromosomes, 1 chromosome with A and 1 with B. So they're going to produce both antigens A and B. So how we're going to illustrate this is we are going to say that somebody with a non-mutant variety that produces antigen, we're going to use the letter I. We're going to say, in this instance, you have an antigen producing allele, right? Whether that's A or B. So we're going to have IA for antigen A, or IB for the antigen B producing allele. And then to show that the mutant allele is recessive to those, we're going to use a lowercase I. So this is the mutant, which is going to produce no antigen. And that is our multiple allele set. So when we look at this, we're going to go ahead and kind of just look at a table, and then we'll sketch out a table. So share this with you. So we look over here, we can see our blood types. We can see blood type A with the A antigen, blood type B with the B antigen, AB with blood types A and B antigens, and then O would be the mutant antigens, or the mutant allele that wouldn't have any antigens at all. So it shows us, again, the antigens present. There's something called antibodies. So if you have blood type A, your body is going to produce antibodies, defense mechanisms against blood type B as a protective measure. And conversely, if you have blood type B with B antigens, your body's going to produce antigens, defense mechanisms against blood type A. So you would have anti-A antibodies. You really don't have to worry about this much right now, but this is why only certain blood types work together in terms of transfusions. Somebody with type O is the universal donor because theirs is kind of a clean canvas and they can donate to anybody. Somebody with type AB can receive any type of transfusion because they have all the antigens possible, so they don't produce any antibodies. Okay, so let's just sketch out, let's go back to our original writing here. And we're just going to sketch this table out. So when you're asked to solve these problems, you've got the ability to do so. So I'm just going to draw a table. And it'll be a pretty simple table. You could probably sort this without this table, but we're just going to be sure. So we'll have blood type A, blood type B, blood type AB, and then blood type O. We're just going to list the genotypes that are possible. So for blood type A, you could have an individual with both of the A alleles, or you could have an individual with one A allele and then the mutant allele, but because the A allele is dominant over the mutant allele, both of these result in a type A blood phenotype. Type B is very similar, where you could have both B alleles or a B allele and a mutant allele and be blood type B. AB, you're going to have both A and B alleles. And then blood type O, you're going to have both mutant alleles. So what we want to do is look at a Punnett square. So let's say that we've got, let's look at an example. I'm going to use a different color. Let's say that we have somebody with type AB blood, and they are going to mate with type O, and they want to know what are the odds they have a particular blood type. So essentially what we're looking for is the F1. So we're going to give these genotypes. Here they are. So we have the AB genotype and the O genotype. Let's set up our Punnett square. We'll just set that up over here to the left. So we'll take blood type O, we'll put that up top. So again, I took this genotype, put it up top. We'll take AB, we'll put this over here on the side. And this is what we end up with. We end up with 50%, 2 out of 4 having type A. So 50% type A. And then 2 out of 4, 50% having type B. So this is how you would execute a blood typing problem. You would have to figure out what the phenotype is and genotype, put that into a Punnett square, and then find the results or the ratios. In this case, the ratio would be 2 type A to 2 type B, or a 50-50 percentage. Okay, so that is multiple alleles, and there's other examples of multiple alleles in the problem set. You'll have multiple allele problem. There'll be one on there that deals with flower color, and it's kind of a tricky problem to set up and solve. But again, it will be a multiple allele problem. So that is multiple alleles. Let's look at kind of the next item on the agenda here. And that is going to be something we call pleiotropy. So number 5. So pleiotropy. And we'll use our example first, and then we'll kind of describe what it is that we're talking about. So let's go and share our screen. So pleiotropy would be kind of this situation where we have one mutant allele, and that provides multiple phenotype effects. So here we're looking at an allele that's been mutated. There's been a substitution. A thymine has been substituted in place of an adenine in this DNA sequence, which results in a changing of the shape of the red blood cells because of this hemoglobin protein. And that leads to a bunch of downstream phenotype effects, okay, including, so again, this mutated allele here can cause blindness, liver failure, or cardiovascular disease, heart attacks. So multiple traits from just one allele. So it's not controlling just one phenotype. It's controlling a whole host of things. So those are the pleiotropic effects. So we'll go back to kind of the notes here, and we'll just put a brief notation on that. That again, you've got a mutant allele. So an allele, so we'll say an allele has multiple phenotype effects, pleiotropic effects. And the example here was the sickle allele. And if you have both sickle alleles, now you can see all those pleiotropic effects, the multiple phenotype effects like blindness, liver failure, heart attack, the things that we just looked at. So it's not just one phenotype, it's a whole host of phenotypes from that mutated allele. Okay, so let's move on and talk about epistasis. So 6. So EPI, stasis. So epistasis is where one gene locus or location alters or impacts a second gene locus or location. So one gene locus alters a second gene locus or location. These traits are epistatic, and we're going to look at an example here. So let's go and look at Labrador retrievers. And labs can have yellow fur, chocolate fur, or black fur. And that is going to be the interaction of two alleles. So we have an allele that allows pigmentation in the fur, and then we have an allele that codes for chocolate colored fur, lips, nose, and eye rings, or black. In order to have that pigment deposited into the fur, you need this first gatekeeper allele in the picture. And I believe it's called melanocortin. And then the second allele over here is called tyrosinase. So in order for tyrosinase, this pigment, to be deposited in the fur, you need a dominant melanocortin gene. So let's talk through this and look through this, because this one can be difficult to kind of master if you don't have a really clear picture of what's happening. So we're going to look at labs, right, which are just dogs. You may have one of these at home, and you can have a black lab, you can have a chocolate lab, and you can have a yellow lab. And this yellow lab can have a black nose and lips and eye rings, or chocolate. Okay, and this is controlled again by two alleles. So the gatekeeper allele for the fur, we're going to say that's the first allele here, is what we call melanocortin. So melanocortin. And this is essentially fur pigment, whether or not pigment can be deposited in the fur. So your dominant allele, your big E here, is one that would produce pigment. Your lowercase e, your recessive allele here, would be the mutant that would not allow for pigment to be deposited. Our second allele, this tyrosinase, then is for the black pigment, whether that's in the nose, lips, eye rings, or the fur. And then the recessive allele here is the brown, so slightly different shaped protein. and we get a slightly different pigment, whether that's in the nose, the fur, the lips, or the eye rings. So we have these two genes interacting with one another. And we're going to set this up and kind of look at this and how this would work out. So let's say that we've got, set this aside. Let's say that we have a yellow lab. So we've got a yellow, we have a yellow lab, and this yellow lab we know happens to have a brown nose. And then we're going to mate that with a chocolate lab. Okay, so let's set this up. Yellow lab, chocolate lab. And our yellow lab, we know does not have one of those melanocortin genes. So it's got two lowercase e's. Because if it did have a big E, it would be chocolate or black. And we know that it's got a brown nose. So that tells us that it doesn't have a tyrosinase dominant allele, that it's actually got two of these recessive alleles, right? Because we could look at this. So again, no pigment in the fur, so we know it's got two of these recessive alleles. And then if it had a dominant tyrosinase allele, it would have black nose. It's got a brown nose, so we know it's actually our homozygote recessive for both. The chocolate lab is going to have at least one melanocortin gene because it's got fur pigment. And then we got little b's here as well because it's chocolate. So this is what we want to do. So we need to set up our F1. And we're going to look at, we'll look at our E's first. So we'll go ahead and put our E's over here. So this is what we get. We get 2 out of 4 that are going to get melanocortin and have pigmented fur. And then we have 2 out of 4 that, depending on what that allele is here that's missing, that we don't know, we could get golden labs or we could get more of the melanocortin, the colored labs. We just don't know yet. Let's go ahead and look at the tyrosinase. And actually, you could, this one's going to be super simple. Right, so we assign our alleles, kind of where they go. One parent here, the other parent's alleles here. Again, how are we doing this? We're separating the E's and looking at those as a Punnett square. Then we're separating the B's and looking at those as a Punnett square because they're on different chromosomes, so they're assorting independently, so we can pull these apart and do this. Every lab here, in this case, is going to not have any of the tyrosinase allele. So they're going to be all chocolate. They're going to have brown noses, lips, and eye rings. And then a portion of them will have brown fur, depending on if the melanocortin gene is there. So we know half of them, 8 out of 16, at minimum, will be chocolate labs. So we'll say at minimum, 8 out of 16 are chocolate labs. Right? So at minimum, 8 out of 16 are chocolate labs with big E, little e, big B, or sorry, big E, little b, little b. So this is a chocolate lab. And then then there would be a possibility that the other half would be yellow labs. So we would know, looking at our offspring, if we get any yellow labs, we would know immediately that this would have to be a lowercase e here. And then we would expect a 50-50 ratio. If we didn't get any yellow labs, that would hedge and make us start thinking that it's likely that this is a big E as well here. So we could do some investigative work and make some conclusions based on the results of our puppies. If we had enough puppies, if we only had 2 puppies or 3 puppies, it'd be harder to make those conclusions. If we had a bigger litter, we would it would be easier to figure that out. So we're just going to kind of leave that ... Because again, if we see a yellow lab in the offspring, we would know what this is. So we're just going to leave that for now. Okay, let's look at the next item. So this would be number 7. So almost finished here. This is polygenic inheritance. So Polygenic, polygenic inheritance. And polygenic inheritance is the additive effect of 2 or more genes on a single phenotype. So we're getting an additive effect. So these are genes, these dominant alleles are compounding on one another. So the additive effect of 2 or more genes on a single phenotype. So let's look at an example to see if we can clear this up. Let's go back over here. And we're going to look at something like eye color. So eye color, the number of dominant alleles you have dictates how dark your eyes tend to be. So in this case, we have this individual here with 4 dominant alleles, in which case they have very dark eyes. Here, this person has 3 dominant alleles. They're a little bit lighter. This person here, 2 dominant alleles, even lighter. 1 dominant allele, even lighter, and then no dominant alleles. Their eyes are extremely light. Then with eye color, we actually have incomplete dominance happening as well, where some of these alleles start interacting with one another and producing these in-between phenotypes. The takeaway here is the more dominant alleles you have, the darker your eyes and eye pigment of the iris tends to be. So that is this additive effect of polygenic inheritance. We also see this with skin melanin, and the number of dominant alleles you have, the darker the melanin is, or the more melanin you have in your skin. There's a continuum here, and we can see that in terms of number of dominant alleles and skin melanin. So let's go ahead and make some notes in terms of that. I'm going to stop sharing the screen. Actually, we're not going to work any problems right now with this. We're just going to write down these examples. So we had skin melanin. And there were like 6 alleles that we saw there that adds up to kind of pigmentation. And then we also looked at eye color, and the more dominant alleles you had, the darker your eyes tended to be. And then there was some incomplete dominance happening there as well. Eye color is actually quite complex. There's a number of genes that are interacting there. It's not as simple as just 1 or 2 genes. Finally, so number 8, almost finished. We're going to look at something called epigenetics. So number 8. And this is what we call EPI, epigenetics. And these are phenotype changes. So changes due to gene expression. So phenotype changes due to gene expression. We're going to go ahead and describe this and look at this on a shared page, just like we've been doing. So let's go ahead and share this. So here we have our DNA, and DNA can be modified by certain groups called methyl groups. And these methyl groups, which is just a CH3 or CH4, they can tag certain regions of DNA and make it inaccessible to transcription factors that are trying to transcribe those genes for later protein production. So you could have changes in development, environmental chemicals, drugs, pharmaceuticals, as you age, or what your diet looks like, that can tag different regions of DNA with certain methyl groups. And that can activate, make genes, switch genes on, or repress genes, switch genes off. And if those are genes that are related to certain important cellular functions, that can be quite impactful in terms of your phenotype. There's these other things called histones, which are proteins around which DNA is wound for compaction and gene regulation. These histones can be modified, where if you put a tag on these histones, that changes the shape. And then now, instead of being nice and loose like this, where these genes are available, accessible, like this pink one, they can be wrapped and wound even tighter with this modified histone, which means this gene is not accessible and transcription factors can't get there. So the binding of these epigenetic factors to these histone tails alters the extent to which DNA is wrapped around the histone and then makes it not available for transcription factors and gene expression. So what does this mean? This means health endpoints like cancer, autoimmune disease, mental disorders, and diabetes can be linked to epigenetic factors that are tied to these mechanisms here. So really interesting component of genetics. So that's all 8 complex patterns of inheritance. I hope that's helpful, and I'll see you next time.